Welcome back guys. Now in this video, let's discuss about the smooth muscle. Now how a skeletal muscle is different from a smooth muscle. Okay, how a skeletal muscle is different from a smooth muscle. See, smooth muscles, they are not voluntary in nature. First of all, they are not voluntary in nature. They are involuntary muscles. They are not under the control of the somatic nervous system. They are under the control of the autonomic nervous system. Means you cannot contract a smooth muscle on will. Next, autonomic nervous system meaning what? Either sympathetic or parasympathetic. Okay, the smooth muscles are under the control of the sympathetic or parasympathetic. Either this autonomic nervous system is going to increase the contraction or it's going to decrease the contraction of the smooth muscles. Okay, depends on the situation. Now, what is the shape of the smooth muscle? The shape of the smooth muscle is a spindle shape with a single nucleus in the center. But what about the skeletal muscle? They are the longitudinal muscles with multiple nucleus. Now, where exactly these smooth muscles are located? See, smooth muscles location, they are located in the organs, visceral organs. For example, intestines. What is the role of the smooth muscles in the intestines? Okay, we know there is a muscularis layer in our GIT. What is the importance of this muscularis layer? That is going to help in the contractions of the GIT. Peristalsis and segmentation helps in the propulsion of the food, helps in the mixing of the food with the different digestive enzymes and helps in the digestion of the food. Okay. Next, in our bronchi, in our airways, the smooth muscles are present. The smooth muscles can lead to bronchoconstriction, bronchodilation. Okay. So that is going to cause the resistance for the flow of air. A severe bronchoconstriction can lead to asthma. Okay. Next, these smooth muscles are also present on our blood vessels. Okay. What is the role of smooth muscles on the blood vessels? They are going to give the resistance for the flow of blood, not the airflow, sorry. It's going to give the resistance for the flow of blood. Okay. See, imagine if a blood vessel is contracted. When a blood vessel is contracted, there is going to be more resistance for the flow of blood. So, the smooth muscles are going to either determine the resistance for the airflow, blood flow, and helps in the movement of the food in the GIT. It helps in the contraction of the gallbladder. It helps in the contraction of the uterus. These are all the smooth muscles which are not under our control. Okay, they are under the control of the autonomic nervous system. See, smooth muscle contractions are considered as slow. They are slow, sustained contractions. We will see why. Okay, we will see why. My dear students, the smooth muscles, okay, there are mainly of two types. Okay, the smooth muscles are mainly of two types. What are the two types of smooth muscles? The two types of smooth muscles are single unit smooth muscle. Okay, single unit smooth muscle also called as a syncytium, or multi-unit. Okay, multi-unit smooth muscles. What is the difference between these single unit smooth muscles or a multi-unit type of smooth muscles? See, there are smooth muscles. For example, let's say there are smooth muscles. Okay, let's say 10 smooth muscles are there. Okay, let me draw it here. Let's draw 10 smooth muscles. Okay, actually smooth muscles are spindle shaped, but I am not showing them as spindle shaped. So just for easy understanding purpose. See, imagine these are all the smooth muscles. These smooth muscles, they are connected with each other. Okay, with the help of these gap junctions. What exactly are they? These are the gap junctions, guys. So now, if one smooth muscle is depolarized, let's say if one smooth muscle, if it is depolarized, now that voltage, the calcium is going to go from one cell into the other cell, from this cell into the other cell, from this cell into the other cell. So all the cells are going to get the depolarization and all of them are going to contract as a single unit, as a syncytium. All are working together. All are going to contract at the same time. This is called as a single unit smooth muscle. That is because of the presence of the gap junctions which allow the voltage to pass from one cell into the other cell. Okay. But what if these smooth muscles are contracting individually? See, this is a smooth muscle which is having a different nerve supply, and here is a smooth muscle which is having a different nerve supply. A different motor neuron is stimulating it. So here the smooth muscles are contracting individually. So they are called as a multi-unit type of smooth muscles. Okay. Now, the examples they will ask you, where do we have such single unit type of smooth muscle and multi-unit type of smooth muscles in our body? Okay. Let me show you here. Say single unit smooth muscles, they are going to help in the contraction, like they are going to contract as a group, and where they are present in the intestines, in the bladder, gallbladder, urinary bladder, okay, uterus. So all the muscles will contract at the same time, okay, causing a coordinated contraction. But where do we see such multi-unit type of smooth muscles? I have already discussed with you. Multi-unit smooth muscles means they are going to contract individually. They are going to cause the fine contractions. So where do we require the fine contraction, individual muscle contractions? In your eye, in your ciliary body, okay, in the ciliary body of the eye, in the iris, in the piloerector muscles. Okay, these are the examples of the multi-unit type of smooth muscles. Now, let me show you the difference between, let me tell you the difference between a skeletal muscle and a smooth muscle. See, in a skeletal muscle, what do we have? We are having a motor units. What does it mean by a motor unit? There is going to be a single motor neuron is going to be there. This single motor neuron is going to innervate myocytes or muscle fibers. But look in the smooth muscle. See, here is the motor neuron, let's say. Okay, here is the autonomic motor neuron. See, this autonomic motor neuron is forming these bulges. Okay, forming these bulges. What are these bulges called as? These are called as the varicosities. And inside these varicosities, what do we have? We have the neurotransmitter. Now, what this neurotransmitter is going to do? The neurotransmitter is going to cause the depolarization of these muscles. And look at here, in between the cells, you are having the gap junctions. Okay, these are the gap junctions. Now, because of the presence of the gap junction, all these smooth muscles are going to contract as a one individual group. That is why it's an example of a single unit, okay, smooth muscle, single unit smooth muscle, which is present in your intestines, gallbladder, uterus. Okay, next. Then what about the multi-unit smooth muscle? See, multi-unit smooth muscle, I have told you, there are no gap junctions. See, this muscle fiber is going to contract individually. This one contracts, this one contracts, this one contracts individually, not as a group. But my question is, where do you see varicosities? The varicosities are not present in the skeletal muscle, but they are present in the smooth muscle. What are varicosities? They are nothing but the bulges in the neuron which contains the neurotransmitter. The varicosities contains the neurotransmitter. Whenever there is a depolarization, they are going to release the neurotransmitter and cause the contraction of the smooth muscles. Okay, done. Next, what else is the difference? Okay, what are the differences between a skeletal muscle and a smooth muscle now? See, in a skeletal muscle, let me tell you, in a skeletal muscle, we have something called as a T-tubules. We know there is something called as a T-tubule. Okay, T-tubule is nothing but the muscle cell membrane, the invagination of the, let's say, this is the T-tubule in the skeletal muscle. Okay, I am talking about the skeletal muscle. See, this is the muscle cell membrane. There is an invagination of the cell membrane into the cytoplasm, that is called as a T-tubule or transverse tubule, on which the DHPR channels will be present. Okay, DHPR channels, okay, voltage gated calcium channels are present, we know. But such T-tubules are absent in smooth muscle. Okay, T-tubules are absent where? In the smooth muscle, there is no such thing as a T-tubule. So, if you look at a smooth muscle, this is how a smooth muscle is going to look like. Okay, just a small depression is there. Okay, there is no well demarcated T-tubule like structure is not present. Just a small depression is present, and here you will have the varicosity, something like this. Now, this small depression is called as a caveoli. This is called as caveoli. And what do we have in the caveoli? In the caveoli, we have voltage gated calcium channels. Okay, which are going to react to the depolarization. These are the voltage gated calcium channels. Okay, voltage gated calcium channels. Okay, the calcium channels which are voltage gated 1.2 type. Okay, voltage gated calcium channels. Next, what about the actin and myosin? We know inside a skeletal muscle there is going to be a proper actin myosin arrangement is there, which is going to give rise to the striation. Okay, that's why skeletal muscles are also called as a striated muscle, because there is going to be a light band, dark band, light band, dark band, A band, I band will be there. Okay, because of the actin myosin arrangement. So let me write here, in smooth muscle, actin, myosin, both are present, but they are not organized, not properly organized. Okay, so what does I mean by? What is that not properly organized mean by? If you look at a smooth muscle, okay, imagine this is the smooth muscle fiber. I am showing you one smooth muscle fiber which is the spindle shape. This is a smooth muscle. Now, here, this is a place, okay, here there is a place from where this actin filaments are coming like this. Actin, 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 actin. And here, there is one more area where these actin filaments are attached. In between these actin filaments, there are myosin filaments present like this. Myosin filaments present like this. And let me show you one more sarcomere, one more sarcomere where again actin filaments are arranged, and here again actin filaments are arranged like this. Now in between the actin filaments, what do we have? Myosin filaments are present like this. Now what I am trying to say, in a skeletal muscle there is a proper I band, A band and striation appearance is there. But here the sarcomeres are not parallel to each other. The sarcomeres are not parallel to each other. The sarcomeres may be oblique to each other. They are not properly organized, so that there is no proper striation appearance. Okay, the striation appearance is not going to be seen here. And one more thing, see, in skeletal muscle we know, in skeletal muscle there is something called as a Z line to which the actin filaments are attached. But any such Z line is present here. See, all these actin filaments, they are attaching to a point which is called as the dense bodies. These are called as dense bodies. So dense bodies are the areas where these actin filaments are attached. There is no Z line, no Z lines in smooth muscle. So tell me, who is not there? First of all, T-tubule is not there. Actin myosin is present but not properly organized, and no Z lines are there. Next, what about the troponin? Okay, in skeletal muscle we know troponin is the one. A troponin is a protein which is going to bind with what? Calcium, especially troponin C. Okay, troponin C component is going to bind with the calcium, and whenever troponin binds with the calcium, that is going to cause the removal of the tropomyosin from the blocking positions. Active sites are exposed, we know. But now in a smooth muscle there is no concept of troponin at all. There is no troponin protein. Okay, so right, troponin is absent. Okay, in smooth muscle. There is no such thing as troponin in the smooth muscle. So now you will be having a doubt, sir, if troponin is not there in the smooth muscle, then who is going to bind with the calcium? Who should bind with the calcium? Actually, there should be a calcium binding protein. So in smooth muscle, the calcium is going to bind with a protein called as Calmodulin. Okay, calcium is going to bind with what? Calmodulin. This is called as a Calmodulin complex. Calcium will go and bind with a protein called as a Calmodulin. So okay sir, troponin okay. Then what about tropomyosin? So you tell me, what is the role of tropomyosin in a skeletal muscle? Tropomyosin blocks the active sites on the actin, prevents the binding of myosin heads with the active sites. Tropomyosin is bad thing. It doesn't allow the muscle contraction to occur because the tropomyosin blocks the active sites so that the myosin head cannot go and bind with the active site. But wait, something paradox here, something unusual here. Tropomyosin, is it present? Yes, it is present, but it enhances, it increases the actin myosin binding. Means the role is exactly opposite. It enhances, it increases the actin myosin binding. Okay, so now you will be having a doubt, sir, if tropomyosin, which was supposed to block the active sites, which was supposed to interfere with the actin myosin binding, which should decrease the actin myosin binding, it is actually promoting it, then who decreases the actin myosin binding? Who interferes with the actin myosin binding? Who hinders the actin myosin binding in the smooth muscle? So for that, there are a different type of protein called as calponin. Okay, in smooth muscle there is something called as calponin and caldesmon. Okay, calponin and caldesmon, what they will do? They decreases, they interfere with the actin myosin cross bridge formation. They decrease, okay, actin myosin binding. Okay, so these are the important differences. One more important difference is there. Anything else structurally that is different from a skeletal muscle from the smooth muscle? In skeletal muscle, we have something called as nebulin, which is an anchoring protein, nebulin, and titin. Okay, in the skeletal muscle physiology, I have discussed about this titin, which is also called as a muscle spring, which maintains the elasticity of the muscle. Titin is the largest protein in the humans. Okay, we have discussed. So titin and nebulin, these two structures are also absent. in smooth muscle. Okay, so these are the differences between the skeletal muscle and smooth muscle. What are the differences, guys? Again, I am repeating, there is no such thing called as a T-tubule. Instead of T-tubule, what we are having, caveoli is present, actin, myosin are present, but they are not properly organized. They are oblique to each other, so there is no striation. And any Z lines are there? There are no Z lines. Instead of Z lines, this is MCQ. Instead of Z lines, what do we have? Dense bodies. Okay, so what is analogous? What is analogous to Z lines in the smooth muscle? Dense bodies. Okay, next, troponin, is it there? There is no troponin. So who is the calcium binding protein in the smooth muscle? Calmodulin. Calcium binds with calmodulin. Tropomyosin, is it there? Yes, tropomyosin is there, but it increases the actin myosin interference. It increases the actin myosin binding. What about the calponin and caldesmon? Calponin and caldesmon, they will, okay, they will block the actin myosin binding. Okay, next, nebulin, titin, these structures are not present. Now let me explain you how the smooth muscle contraction is going to occur. Okay, how the smooth muscle contraction occurs. Let's say this is the cell membrane of the smooth muscle where there is a depression called as a caveoli. Okay, from the varicosity, the neurotransmitter is released, acetylcholine is released, or norepinephrine is released. Let's say for now, this muscle is getting depolarized. For the first time, the smooth muscle is getting depolarized because a neuron is stimulating it. Okay, a neuron from the varicosity, it is stimulating it. So now you tell me, whenever this depolarization is spreading along the smooth muscle membrane, now the depolarization will come to the caveoli also. Now, in the caveoli, you tell me which type of channels are present. Sir, in the caveoli, there are voltage gated calcium channels, type 1.2, they are present. Now, for the first time, because of this depolarization, see, because of this depolarization, these channels will open, which allow the calcium to come into the cell. Okay, the calcium is coming into the cell. For the first time, the extracellular calcium comes into the cell. Okay, now where do you think this calcium is going to go and act? This calcium, it will go and act on the smooth endoplasmic reticulum. So inside the smooth muscle, there is smooth endoplasmic reticulum. Okay, now on the smooth endoplasmic reticulum, there is one more channel present. There is one more channel present. So look at here. This channel is called as RYR channel, ryanodine receptor type 3. Okay, so ryanodine receptor type 3 is present over here. Now this calcium, it will come and stimulate, the calcium will come and stimulate these ryanodine receptors. Whenever the ryanodine receptors are stimulated, actually the calcium is going to open these ryanodine receptors. So who is going to be released? The calcium will be released from the sarcoendoplasmic reticulum. Okay, so from the sarcoendoplasmic reticulum, the calcium will be released outside, because we know the sarcoendoplasmic reticulum is a store of calcium. So this is how the calcium is available for the smooth muscle contraction. Now can I call it as, see, electro means because of the electrical activity. See, you tell me what exactly happens in the skeletal muscle. See, in skeletal muscle there is something called as a electromechanical coupling. That is because of the depolarization, DHPR channels are going to open the RYR channels in the skeletal muscle. The skeletal muscle, the DHPR channel will come and open the RYR channel mechanically. That is the electromechanical coupling. But here in the smooth muscle, what exactly is happening? Sir, whenever there is this depolarization or action potential, these calcium channels will open. So calcium is going to come into the cell. So it is this calcium which is going to induce the release of this calcium. So this is called as calcium induced. Calcium release. Okay, in the short form you can call it as CICR, calcium induced calcium release, which is also seen in the cardiac muscle. Calcium induced calcium release is in cardiac muscle as well as the smooth muscle also. But in skeletal muscle, it is electromechanical coupling where the DHPR channel is opening the RYR channel. Already I have explained you that. Okay, now you tell me what this calcium is going to do. What this calcium is going to do? Now this calcium, it is going to come and bind with a protein, calcium binding protein is called as what? Calmodulin. Okay, calcium is going to bind with calmodulin. So this is called as, now this is called as a calcium calmodulin complex. Okay, calcium calmodulin complex. Now, what is the role of this calcium calmodulin complex? The calcium calmodulin complex is going to activate something called as myosin light chain kinase. Okay. So this calcium calmodulin complex is activating an enzyme. It's actually activating an enzyme called as myosin light chain kinase. So now you tell me, okay, now you tell me what this myosin light chain kinase is going to do. Okay, kinase means what? It will do the phosphorylation. It is going to cause phosphorylation. Phosphorylation of what? Phosphorylation of what? Students, look at here. This is the myosin, let's say. Okay, here is the myosin. And here is the actin, let's say. Okay, here are the actin filaments. These are the active sites. See, in smooth muscle, here is the myosin head. Okay, the myosin head. You tell me the myosin head here, it is not properly oriented. It is in a bent form. Okay, the angle, see, the angle is very, very less. Now, what we need to do? See, here there is a myosin light chain. These heads are made up of myosin heavy chains and light chains. Now, what we need to do is this enzyme myosin light chain kinase, it will come and it will cause the phosphorylation of the myosin light chains. Because of this phosphorylation, the myosin head angle is going to increase. Now the myosin head will become like this. The angle is going to increase. Now this is ready. Okay, now it is ready to go and bind with. Now it is ready to go and bind with the active site. Now it will go and bind with the active site and it's going to cause the pulling of the actin filaments. So muscle is going to contract. So now tell me, what is this MLCK? That is myosin light chain kinase is doing. The myosin light chain kinase is going to increase the angle of the myosin head so that it will go and bind with the active site and cause the muscle contraction. Okay, contraction. Not only that, this myosin light chain kinase is also going to increase the ATPase activity, increase angle and also increase ATP is activity in the myosin heads. Okay, so more ATP will be broken down. So the ATP is activity and the angle is dependent on the myosin light chain kinase. So this is the super important enzyme. Now the same enzyme should have to do one more thing. What is that? In smooth muscle, I have told you just increasing the angle is not enough. Who is trying to prevent this binding? Who is going to prevent the binding of actin from myosin? In smooth muscle, desmon and calponin and caldesmon is trying to prevent the binding of actin and myosin. So the same myosin light chain kinase is going to inhibit or it is going to do the phosphorylation of calponin and caldesmon. Okay, calponin and caldesmon. So what's going to happen? Whenever this calponin and whenever this caldesmon, whenever they are phosphorylated, they are going to not interfere. They are not going to interfere with the actin myosin binding. So now the myosin can go and bind with the actin active site. So muscle contraction is going to happen. Okay, muscle contraction is going to happen. So now tell me, what are the two important events of this myosin light chain kinase? The myosin light chain kinase is going to increase the angle, increase the ATPase activity, but the same myosin light chain kinase is also going to inhibit the calponin and caldesmon. Okay, so muscle contraction will occur. Okay, and tell me how muscle is going to relax. How this muscle is going to relax? So what you need to do? Again, there is one more enzyme called as myosin light chain phosphatase. Okay, MLCP, myosin light chain phosphatase. Now what this myosin light chain phosphatase is going to do? The myosin light chain phosphatase is going to dephosphorylate. Okay, kinase is doing the phosphorylation. This is phosphatase. So what it will do? It is going to cause the dephosphorylation so that now the angle will go down so that it is no longer going to bind with the active site. So for contraction, you require myosin light chain kinase, and for the relaxation, you require the myosin light chain phosphatase. Okay. Now, apart from this, what else you should know regarding the smooth muscle contraction? Very important point is there. That is, look at here. The contraction of the smooth muscles does not require action potential or depolarization. The muscle contraction, the smooth muscle contraction, not always require the depolarization. Even without depolarization, the smooth muscle can contract. Now you will be having a doubt. How, sir? How is it possible? Let me tell you. Guys, look at here. Let me draw the smooth muscle again. This is the smooth muscle cell membrane. This is the caveoli. Okay. Caveoli. Now, here is the smooth endoplasmic reticulum where there is store of calcium. Where there is store of calcium. Okay. Now, what I am trying to say, see, I am not talking about any depolarization. Now, I am not talking about any depolarization. Now, see, on the smooth muscles, there are some G protein coupled receptors are present. Okay. There is a G protein coupled receptors. Some receptors are present. Even drugs. Okay. So, certain drugs can actually come and stimulate this G protein coupled receptor. So, whenever this G protein coupled receptor is activated, they are going to produce something called as the IP3, secondary messenger called as the IP3, inositol triphosphate. Now, what is this inositol triphosphate can do? The inositol triphosphate, it can come, it can stimulate this endoplasmic reticulum. Actually, on the endoplasmic reticulum, there are receptors. Okay, there are receptors for inositol triphosphate, receptors are there. Now, this inositol triphosphate is coming and stimulating the receptors so that again calcium can be released, and you know the story, the calcium will come and bind with Calmodulin, and this calcium Calmodulin complex is going to activate myosin light chain kinase, and myosin light chain kinase is going to help in the muscle contraction. So, what I am trying to say here, there is no need of depolarization or action potential for the actual contraction of the smooth muscle. Okay. So, pharmacological agents like drugs can even cause the muscle contraction. Okay. This is called as a pharmacological coupling. Okay, pharmacological coupling. See, in skeletal muscle, what we have seen, it is the electromechanical coupling leading to the muscle contraction. Here, it is a pharmacological coupling for the smooth muscle contraction. So, pharmacological coupling is seen in, okay, the smooth muscle contraction. Action potential is not needed for the contraction of smooth muscles. Okay. Now, anything else that you should know regarding the smooth muscle? There is something called as a latch bridge phenomena. Latch bridge phenomenon. So, what exactly is this latch bridge phenomena? Latching means what? Latching means holding on. See, what I am trying to say, during smooth muscle contraction, during smooth muscle contraction, the myosin head is going to go and bind with the active site. Okay, the myosin head, it will go and bind with what? The active site, and causing the muscle contraction. Okay. And during relaxation, the myosin head will detach. And it's going back to original position. But in smooth muscles, see, in smooth muscles, even after the relaxation, during the relaxation phase, some cross bridges are still present. The myosin head is some cross bridges, they are just like that. Okay, they are not going to detach. The myosin head is not going to detach with the active site. Okay, let's say if there are 100 cross bridge formations, so some cross bridges are just left over like that. Okay, there is no complete removal of the myosin from the active site. Why? Because the smooth muscle is still in contracted phase. Okay, just like that. Okay, even without ATP, there is a sustained contraction of the smooth muscle. So, the muscle is contracting, okay, even without using the ATP, just like that, the muscle tone is maintained. So, why this is important means the muscle contraction even without the ATP, by using less ATP. So, where do we want such latch bridge phenomena? In the smooth muscles. Why? To properly maintain the tone of the smooth muscles. Okay, in our body, in order to maintain the BP, the smooth muscles always need to be contracted, and there should be a certain degree of contraction. So, for that contraction, I can't use much energy, 24/7, I can't use the energy. So, in the vascular smooth muscles, let's say for example, in the vascular smooth muscles, even without ATP, okay, the vascular smooth muscles, they are always in contraction phase. Even after the relaxation phase also, these heads, they are still binding with the active sites and the muscle is still in a contracted phase. So, contraction is happening, maintained even without the ATP. So, that is called as latch bridge phenomena, which is seen in the smooth muscles. Okay. So, with this, all the important points about the smooth muscle are completed. Let me highlight the important points again. You already know it. See, what are the important points, guys? Troponin, nebulin, titin. They are all absent in the smooth muscles. True. Calcium is going to bind with what? The calcium is going to bind with Calmodulin. So, which is forming the calcium Calmodulin complex. Tropomyosin, is it present? Yes, present. But it promotes the actin myosin interaction. It promotes. What about the calponin and caldesmon? Calponin and caldesmon, they inhibit the actin myosin interaction, true. Okay, only phosphorylated myosin light chain is going to interact with the actin. Means if you want the myosin to go and bind with, the actin, the myosin light chain need to be phosphorylated with the help of enzyme called as myosin light chain kinase. Phosphorylation is going to help in contraction, dephosphorylation is helping in relaxation. Phosphorylation is done by myosin light chain kinase, dephosphorylation is done by myosin light chain phosphatase. Okay, phosphatase. I have told you, look at here my dear students, see it's the myosin light chain. Okay, so with the help of this myosin light chain kinase, there is going to be contraction, and myosin light chain phosphatase, it's going to cause the relaxation. It's a cycling, the cycling is happening. Okay, the two important enzymes which I wanted to know for your exam. Okay, I have told you, see this G protein coupled receptors. The G protein coupled receptors can produce the inositol triphosphate, and the inositol triphosphate, what it will do? The inositol triphosphate, it can stimulate the release of the calcium, and the calcium forms the calcium calmodulin complex. The calcium calmodulin complex activates the myosin light chain kinase, and you know the story. Muscle contraction is going to happen. Okay. So with this, successfully we have completed the smooth muscle physiology. Hope the video is helpful. See you in the next video. Thank you.